Hello, and thank you for your interest in the BSc in Molecular and Translational Haematology. My name is Jim Crawley, and I'm a Professor of Haemostasis in the Hammersmith Hospital campus at Imperial College, and I'm the Director of this BSc course. Although so far in your medical degree you may only have had comparatively small amounts of pure haematology, you will certainly be familiar with many of the themes and principles that run through this course. Over the next few slides, I will endeavour to give you a bit of a flavour of the Molecular and Translational Haematology BSc, the course content, and its broader relevance to clinical practice. To begin with, the first and perhaps most important question is, what exactly is haematology? Well, in general terms, this encompasses the study of the cause, prognosis, treatment, and prevention of diseases related to the blood. These diseases, as you will see, include a wide variety of disorders, including genetic deficiencies, cancers, autoimmune diseases, and also cardiovascular disorders. Perhaps the second most important question to answer is why you should, as a medical student, study haematology. First, due to the importance of the blood and blood cells in so many essential physiological but also pathophysiological processes, haematology has relevance to just about every branch of medicine. Therefore, haematology provides essential training for all clinicians, irrespective of the branch of medicine that you may ultimately specialize in. Second, haematology as a discipline is clinically very important, as it encompasses topics and diseases that afflict a very large number of patients, as you appreciate in the slides that follow. The course covers diverse and varied themes that are also pertinent to many medical disciplines, including but not limited to stem cell biology and cell development, immunology and autoimmunity, so leukocyte function and different examples of diseases associated with the consequences of our immune system acting on our body's own cells or proteins. Cancer biology, how certain cancers develop and how they can be treated. And cardiovascular science. It's worth noting that cancer and cardiovascular diseases together account for over half of all deaths. The treatment of hematological diseases is varied, but includes pioneering cell-based therapies, including stem cell transplant and CAR T-cell therapy, gene therapy, and indeed gene therapy for haemophilias offers the potential to now cure genetic deficiencies in factor VIII or IX. We also cover novel immunotherapies and also rationally designed drugs that specifically target cancer-causing proteins. Module 1 of the Haematology BSc is split into 3 blocks that cover diseases of the white blood cells and bone marrow, which encompasses leukemias, lymphomas, and myelomas. Block 2 covers diseases of red blood cells, including sickle cell disease, various hemoglobinopathies, and red cell enzymopathies. And Block 3 explores disorders of platelets and plasma components, including platelet disorders, coagulation deficiencies such as haemophilias A and B, as well as the very common thrombotic disorders including heart attack, stroke, and venous thromboembolism. In Block 1, you will learn about stem cell biology and hematopoiesis, which covers the basic biology of how the different blood cells develop, which itself is essential for understanding how bone marrow and white cell disorders are manifest. This block then covers what happens in different types of leukemia, including CML, CLL, AML, and ALL. Together, these leukemias represent about 3% of all cancers. A major success story in the field of leukemia was the identification of the chromosomal translocation in chronic myeloid leukemia, or CML, that causes the formation of the Philadelphia chromosome and the cancer-causing BCR-ABL fusion gene. In the 80s and 90s, the 5-year survival for CML patients was only about 20 to 30%, highlighting how deadly this disease can be. The development of inhibitors that specifically target the fusion protein kinase that causes the disease have transformed the treatment of CML, and in combination with the highly improved monitoring of this disease, nowadays means that CML patients have a near normal life expectancy. Despite the progress in CML, the prognosis for other leukemias is not quite so good, which remains an ongoing challenge for clinicians and scientists alike. Block 2 also covers lymphomas. These are cancers that develop from lymphocytes, and also myelomas, which are caused by the expansion of abnormal plasma cells. The mortalities associated with these diseases remains comparatively high, highlighting the importance of further scientific and clinical research into these areas to improve management and treatment of these patients. Block 2 in the Molecular and Translational Haematology BSc delves into the biology of red cells and red cell disorders. This encompasses the normal development of red blood cells. You will then learn about the pathophysiology of different red cell disorders, including hemoglobinopathies, thalassemias, and sickle cell disease, how these disorders are manifest and treated. Both of these diseases have high prevalence and as such are of particular clinical importance. Finally, in Block 2, students will explore the red cell membrane enzymopathies such as G6PD deficiency, which is the most common inherited human enzyme deficiency, affecting an estimated 400 million people worldwide, and also pyruvate kinase deficiency. Both of these enzymopathies can cause or contribute to the onset of hemolytic anemias. Module 1 will also cover some of the different cellular therapies for diseases of the bone marrow and blood cell cancers, including bone marrow and stem cell transplants, as well as some of the practical challenges and potential side effects associated with transplantation. This block also covers the rapidly developing and pioneering CAR T-cell therapy. This involves the isolation of a patient's T cells. These cells are then reprogrammed so that they can better target cancer cells, and the expanded cells are re-injected back into the patient. This therapeutic approach has the potential to transform the treatment of many different types of cancer. Block 3 of the Molecular and Translational Haematology BSc covers hemostasis, or blood clotting. Blood clotting, as shown in this video, involves the specific recruitment of blood platelets, shown in green, to the site of vessel damage. Thereafter, the coagulation cascade catalyzes the deposition of fibrin, shown in red, that holds these platelets together. In Block 3, you will learn about the biochemical and cellular basis of blood clotting and the processes that lead to the formation of a clot, shown here in this electron micrograph. In this image, you see platelets, shown in grey, held together by the intricate fibrin strands, shown in orange, that together prevent bleeding. Armed with this information, you will learn how patients with either inherited or acquired deficiencies in components of the hemostatic system can suffer from lifelong bleeding disorders such as von Willebrand's disease, haemophilia, and immune thrombocytopenia, and the innovative ways in which science has developed therapies for these patients. Perhaps of greater clinical importance both to society and the healthcare system are the effects of excessive clotting that causes thrombosis, including myocardial infarction, ischemic stroke, and venous thrombosis. Importantly, thrombotic diseases account for about 1 in 3 deaths worldwide, and as thrombus formation is the event that kills people, this represents the major modifiable endpoint. Therefore, understanding the molecular and cellular basis of hemostasis not only informs us about the pathogenesis of thrombotic diseases, but it also provides the opportunity to exploit this to intervene therapeutically. Anticoagulant and antiplatelet therapies save millions of lives every year. The Molecular and Translational Haematology BSc is organised and delivered by international experts in the field. During Module 1, we deliver the course content that I've outlined in varied ways to try to enhance both learning and the student experience. We employ a mixture of flipped classrooms, lectures, and discussion sessions to augment student understanding. Over the last couple of years, we have delivered much content online, and we have adapted our teaching to accommodate this. However, we are well set up to deliver teaching both face-to-face and online, depending on the government and university guidelines. During the last academic year, we provided a blended approach of both face-to-face and online teaching. This year, we have also held coffee mornings and informal sessions to provide a forum for discussion of the course, its contents, and any ongoing issues. For the in-course assessments that we have, we hold formative assessments and Q&A sessions to help guide the students through the different assessment modalities. Module 2, like some of the other BSc courses, involves a clinical case study, and this includes attendance at haematology clinics as part of the Science in Context components. And there is also a group literature review, which is performed in groups and is an incredibly popular component of the course. Module 3 involves the 15-week research projects. In haematology, we offer a mixture of both lab-based and clinical research projects. On this slide, you can see some of the different projects that have been offered over the years. In green, you see examples of lab-based projects, and in blue, the clinical projects. I will finish here. In closing, I would like to remind you that the Molecular and Translational Haematology BSc is not just for prospective haematologists. Irrespective of your future clinical specialty, this course covers topics that are essential for and will benefit all clinicians. The Haematology BSc will both inspire and challenge you in equal measure, and it will provide a direct link between basic science and clinical practice. Please visit our website for further details, as well as videos and testimonies and entry requirements. And if you have any further questions, please don't hesitate to contact me by email.